Hello. Today we're going to be discussing Bacillus thuringiensis and Streptomyces hygroscopicus, two bacteria that are commonly used in genetic technology. First, we'll be discussing Bacillus thuringiensis. Bacillus thuringiensis is a member of the phylum Bacteria. It's gram-positive, which means that it produces a peptidoglycan coat. If you don't know, a gram stain, in a gram stain, the bacteria are stained with crystal violet. The crystal violet works its way into the peptidoglycan coat, so that if the bacteria does produce a peptidoglycan coat, it looks reddish, and if it does not, it looks pinkish. It's a very convenient way to classify bacteria and tell between them. This bacterium lives in soil, in the dark parts of leaves, and in caterpillar guts. It is, like all other members of its family, aerobic, and it produces endospores. Endospores, if you don't know, are a type of very, very strong shell in which some genetic material in cytoplasm is contained, so that the bacterium can continue living even after incredibly strenuous environmental fluctuations. So, for instance, if you heat chop the bacterium, you might not destroy the endospore, and the bacterium can continue reproducing after the strenuous conditions pass. This is what Bacillus thuringiensis looks like. As you can see its characteristic rod-shaped form, and it's quite small. The bacterium was discovered in 1902 by Shitanagane Ishiwatari in Japan, and then it was rediscovered in 1911 in Germany. It turns out to be related to Bacillus cereus and Bacillus anthracis. As you may know, Bacillus anthracis produces the incredibly deadly disease anthrax in humans, and Bacillus cereus has many different strains, some of which produce serious problems, and some that others are simply not pathogenic at all. The main difference between these species are the plasmids that the bacteria contain. For instance, thuringiensis has a plasmid with a class of genes called cry genes that Bacillus cereus does not have. Cry genes are genes that allow Bacillus thuringiensis to produce a class of proteins called delta endotoxins. It produces so much of these proteins, in fact, these proteins form crystal masses in the bacteria. These proteins are toxic to certain species of insects. They're incredibly specific to certain species, so that the proteins basically have no effect on any species except the ones they do affect. The way they work is the alkaline pH of insect guts activates the proteins, and that causes the gut cells to lyse, which stops the insect's digestive system, and the bacterium dies of starvation. The insect dies of starvation. This insecticidal activity is incredibly useful and has been used a lot in agricultural science. For instance, the bacterium is used as an insecticide in spray form. The delta endotoxins have been purified, and they're also used as an insecticide in spray form. And most interestingly, fairly recently, these cry genes have been cloned into plants to produce genetically modified plants using agrobacterium. And this way, this eliminates having to spray insecticide everywhere. The plants just produce their own insecticide, making them more resistant and hardier.
And the other bacteria is Streptomyces hygroscopicus. This is a picture. Streptomyces hygroscopicus, like all other bacteria in its genus, is gram-positive. It's commonly found in soil and decaying vegetables, and where it works as a decomposer. And its structure, curiously, resembles fungi. Its cells are arranged filamentously and branched, and it forms mycelium. And the genus, members of the Streptomycin genus are used to make antibiotics, and the hygroscopicus is commonly used to make the antibiotic rapamycin.
Strangely, there isn't really a lot of information about this particular species of bacterium on the internet. All right.
So yeah, to say the last bit.